All right, let's jump right into this explainer by confronting a pretty brutal truth about the Cambridge Biology 9700 exam. Believe it or not, 9 out of 10 students are losing incredibly easy marks because of just one fundamental error. We actually call this the cold open mistake. It's when you sit down, flip open the paper, spot a familiar topic, and just immediately jot down some generic fact you've memorized. But here's the secret, right? Examiners aren't just testing what you know. They are testing exactly how precisely you can apply that knowledge to the specific data right in front of you.
Take a look at this comparison. It's kind of crazy how fast a flawed generic answer completely collapses when you stack it up against a top-band response. Notice that red pen annotation on the left. The diffusion was slower because the block was bigger. I mean, sure, it sounds biologically true, but that earns literally 0 marks in an A-level context. Why? Simply because it lacks precise, context-specific data. The top-band answer with the green check doesn't just casually state a fact. It uses comparative connectives and pulls specific data points directly from the experiment to build a completely bulletproof argument.
So, how do we avoid that dreaded red pen? Well, we're going to use this 4-step procedural framework to elevate any response from just a simple observation to a structured, data-driven argument. Honestly, this is your ultimate high-mark exam strategy. Step 1. Identify the core biological concept. Step 2. Extract specific evidence and data from the materials you're given. Step 3. Apply your scientific reasoning. Really explain the why behind that data. And finally, Step 4. Lock in your answer using precise examiner wording and command words.
Okay, let's look at case study 1, surface area to volume ratio, and how we apply this framework to diffusion rates. Now, this is one of those classic topics where it is so easy to drop points if you aren't perfectly careful. The absolute most crucial point here is mastering this core biological principle before you even glance at whatever experimental data the examiner throws your way. The theory is strict. As cell size, meaning volume, increases, the surface area to volume ratio decreases. This mathematical relationship directly impacts diffusion rates across membranes. If you don't lock down that exact causal relationship first, your analysis of the data is going to fall apart faster than a cell without a membrane.
Notice how the top-scoring candidates don't just rely on the theory. They extract highly specific evidence. Just look at the agar block data here. A top-band answer explicitly points out that block C, which is 30 millimeters, diffused only 20%, whereas block A, at 10 millimeters, diffused 50%. You absolutely must pair your theoretical concept with those exact numbers. And check out the method mark strategy right in the middle. This is a huge takeaway. Examiners will often award method marks just for showing the correct procedural process, even if your final numerical calculation is a bit off. So never, ever skip showing you're working out. It literally saves grades.
Moving on to case study 2, the Bohr shift. We'll be navigating unfamiliar contexts and graph analysis.
Stepping into another notoriously tricky topic, we are going to apply that same deconstruct, solve, evaluate rhythm. Before you even try to interpret a graph or do a calculation here, you have to define the precise causal relationship happening inside the bloodstream. The Bohr shift dictates that an increased carbon dioxide concentration lowers hemoglobin's affinity for oxygen. Visually, this means the oxygen dissociation curve shifts to the right. If you just write something like more CO2 makes oxygen fall off, the examiner is bringing out the red pen for a superficial description. You need that precise terminology.
Just look at this graph tracking the Bohr shift mechanics. The flawed response completely omits the data and the units. To actually secure those marks, like you see with the green check mark, you have to pull specific data right off those curves. You need to state that at a partial pressure of 10 kilopascals of oxygen, hemoglobin saturation falls dramatically from 80% on the normal curve to 60% on the shifted curve. Identifying the data point is a good start, sure, but including kilopascals and percentages, those exact scientific units, is what actually gets you the mark.
Next up, the interactive challenge, the 10-second countdown. All right, now it is your turn to play Senior Examiner. Keep your eyes peeled for generic terminology that totally ignores the specific stimulus material required by the 9700 syllabus. I'm going to show you a beautifully detailed animal cell. A student has labeled a specific organelle, but they've fallen straight into a classic middle school biology trap. Do you think you can spot the error before the timer runs out?
Okay, did you spot the label on that organelle? Yep, the student literally wrote powerhouse. If you caught that generic term instead of explicitly stating mitochondrion, site of aerobic respiration and ATP synthesis, well, you just saved yourself a critical grade.
The 9700 syllabus absolutely demands specific high-level terminology. Phrases like powerhouse or control center are going to instantly lose you marks. You have to use the exact right terminology for the biological situation.
Let's wrap up with our top examiner warnings. Red pen alerts, straight from the principal examiner reports.
To make sure this all really sticks, you've got to avoid these 3 fatal traps that examiners are constantly highlighting in their reports. Number 1, generic, vague answers. You absolutely must tailor your response to the very specific scenario given in the question. Number 2, terminology and units. Double-check your scientific units every single time you write them down. A massive, massive trap is confusing the amount of a substance with its concentration. They are totally different things. And finally, number 3, evaluation failure. Whenever a command word asks you to compare, examiners are requiring you to provide both justified similarities and differences fully supported with evidence. Just doing one or the other will totally cap your grade.
So I'm going to leave you with this. Are you going to risk losing marks to the cold open mistake? Or are you ready to use this 4-step framework to transform your biological knowledge into an undeniable top band response? I highly challenge you to apply this structured strategy, especially the data extraction and unit checks, to the very next past paper question you do. Keep pushing for that precision, and I'll see you in our next explainer as we conquer even more Cambridge biology secrets.